guys. Again, welcome. Welcome to our Bio 3-4 lecture. So we are in the second year of the study design. So the study design was new for 2022. Hopefully you are a little bit familiar with it. Again, if you guys did Bio 1-2 last year, then you will be in, I guess, like the same study design, which is good. At least there's not that kind of change over. In terms of how this lecture will work, so we've got two content blocks. You can see they're kind of divided up along the bottom. So we'll do most of our area study 1 stuff, and then we'll have a bit of Q&A and then a break, and then we'll do our area of study 2 stuff as well, and then a bit of Q&A at the end. In terms of these slides, the recording of the lecture, they'll be able to be accessed a little bit later on. And hopefully if you've guys seen, there will be like a little Slido question, little box where you can pop in any questions that you've got. You can also see other people's questions, so you can feel free to just upvote them if you've got the same question as them rather than asking the same one. But again, if you don't see it, that's fine, just pop it in. So yeah, we'll have a few Slido polls as well throughout the lecture, so we'll kind of do some stuff on there as well. But yeah, please feel free to ask questions. Again, we will do a bit of Q&A at the end of the first content block and then at the end of the second one. So while I'm going through the slides, if you've got questions, please ask them. I won't be stopping, like obviously in between to answer them, but we'll answer them in a big Q&A block, again, halfway through and at the end. Okay, in terms of who I am, as I mentioned, my name's Loz, so I tutor Bio 3-4 at TuteSmart. I graduated in 2020, so I'm going into my third year of uni now at Monash, and I got a 45 in Bio as well. And I did Bio when I was in 2020, so when I was in year 12. So I did Bio 1-2 in year 11, and then Bio 3-4 in year 12. Okay, so in terms of some sort of quick tips for Bio, I know we'll get to a Slido poll in a bit about how you guys are feeling about it, but Bio can have this kind of, I guess, scary perception, like I feel like some students might have. I know when I went into Bio 3-4, I was a little bit like, ooh, I kind of heard things, it's like really hard, it's content heavy, stuff like that. But you just have to approach it in the right way and you'll be able to be successful. So in terms of some tips, the study design, especially for you guys, because you are in the second year of the study design. So it's really important, particularly when you're going through old resources, to understand which questions are still relevant and which aren't, and also to understand what's been added to the study design and what's different from the last one, just so you're aware that you may not see as many questions on that kind of stuff, if that's what makes sense. Getting ahead, in terms of like getting ahead, it does depend on the individual, like how far ahead you want to get. By this, I kind of mean just like have that view of the study design and know what's kind of coming up, but not necessarily, like you don't have to cover the content specifically and really in depth before you're taught it. It is up to you. I never kind of like went ahead on the content from class, and that was solely because of the type of learner that I am. But I know, I had friends who went ahead and that obviously helped them and worked for them. I know for me, like I tried to do it in some of my other subjects and it just didn't really suit me. But the main thing is do not fall behind. Try your best not to fall behind because there is a lot. So as long as you've got an idea maybe of what's coming up ahead in, 2 weeks time, or what the next area of study is before you're there. And so you've got a bit of a baseline, rather than, if you're being taught it for the first time and you've never heard of these concepts, you almost have to, it makes you probably a little bit more likely to fall behind. And that's what you want to avoid this year. Practice questions, a huge thing for Bio. For me, Bio, out of all my other subjects, practice questions were the most important. They're important in like all of your subjects really, but super vital for Bio. So really do those. Obviously, when you correct your practice questions, kind of learning from that, asking questions, whether it's to your teacher, to your friends, to your tutor, anything like that, just to make sure that you have a thorough understanding, because with Bio, and especially short answer questions and stuff like that, you need to have an awareness of the entire concept. At the same time, with the last two, I know I've kind of been talking about tips, and obviously you want to get a good foundation in January or in February. Understand that you don't want to get too stressed out as well. You don't want to burn yourself out. Take your breaks. Think about obviously your goals, but don't get too overwhelmed and don't, bite off more than you can chew as well. So hopefully that kind of helps a little bit. in saying that, with some of those tips, we will go to a bit of a Slido poll now. And it's just a general thing of what you guys are a little bit worried about in terms of going into bio this year. And I can see there are a couple of questions already. We'll get to them at the end of the content blocks. I do want to say as well, there are a lot of slides in this lecture, and obviously bio is super content heavy, as I've just been talking about. I do want to prioritize time for questions because, again, you guys will get a copy of these slides. So if towards the end of the content block, like I'll try to go as fast as I can without rushing, obviously. But if at the end we're a little bit pressed on time, I will answer the questions as opposed to trying to finish everything. So if we don't get to the end of, like, I think cellular respiration is our last little thing to do, we may not cover all of that, you know, again, just due to time. So I will prioritize the Q&A. But if anything, you guys will get the slides, so you'll be able to look through it, and you can email me if you've got any questions as well. Hopefully that makes sense, because I just feel like it will be more worth your time for me to answer the questions as opposed to never seeing them than it will be to talk through slides that you're going to get. If that makes sense. But yeah, we'll get to that when we need to. Okay, so yeah, keep writing stuff. So I'm seeing content, exams, SACs, yeah, complex content, yeah. The poster sucks, I'll just say that. Research methods and like scientific investigation is challenging, and I urge you guys, do not neglect it throughout the year. It's very easy, especially during exam time. SAC prep as well, I don't know, it depends. Like your teacher will kind of tell you how much, hopefully, like scientific stuff will be in it. But especially towards exam time, it's very easy to always prioritize the content, because sometimes you think, like, you know, scientific stuff, like your variables and your, you know, errors or whatever, like that stuff, you apply it, you know what I mean? So if you have an experiment and it's about photosynthesis, obviously you have to have good knowledge about photosynthesis, right? And then so people will sometimes prioritize content but neglect the actual like scientific investigation stuff. Do not do that, because then you're going to get a photosynthesis question and you'll know all about photosynthesis, but you won't know how to answer the questions that are actually about scientific method and stuff like that. So try your best. Don't fall into that trap of ignoring scientific investigation because you think like you just need to know the definitions and you'll be able to apply it. You have to regularly practice that stuff and revise it. So yeah, content, exam, SACs, balancing subjects. Yeah, definitely. Not doing unit 1, 2. Yeah. Okay. In terms of doing unit 1, 2, I guess it helps a bit. I mean, like, I feel like it's more of a confidence thing, and maybe that idea of having some foundation in science, like, especially with the scientific investigation, I feel like that's the most relevant part, because the scientific investigation stuff you learn in 1, 2, that's what carries over. However, it's not like, having not learned that stuff in 1, 2, like you'll never be able to get it in 3, 4. It's not like that at all. Yeah, there's nothing in 3, 4 that is like vitally taught in 1, 2 that isn't taught again in 3, 4, if that sort of makes sense. So don't feel like you will be behind. You may just need to cover things a little bit more, especially because, again, if you've learned it in 1, 2, then you'll have it in the back of your mind. So you may just need to go over a couple of things, but it's not really any reason for you to fall behind. I know, like, at the school I went to, I did it in 3, 4. So many year 11s did it, and obviously without the 1, 2, like they didn't do the 1, 2 in year 10. And yet, like, they were fine. They did well. So don't be too worried about that, I guess. You're not at a disadvantage or anything. Super important, reading the question properly and actually answering what the question is asking accurately. With bio, there's such a specific way of answering short answer questions. And I think often where people trip up or lose marks is this idea of not reading the question properly, or assuming what they think the question will be about, and then it's incorrect. Content, yeah. Studying, yep. Online, yep. Okay, I'm just trying to read this. Yeah, so I'm seeing a lot of stuff with content and kind of balancing. Scientific poster again. Anki, yeah. So Anki, I didn't use Anki until uni, and even now I have a love-hate relationship with Anki. I personally didn't use flashcards and stuff like that in year 12. Um, but yeah, just because for me, I didn't really like them, but I know they're really helpful for other people. A lot of people that I go to uni with, like they, like Enki is their, like, I don't know, their whole life. Like they really, it helped them a lot. And so that's why I'm kind of trying to use it a little bit more because I know it is really, really, really helpful. So if you think it might be for you, if you just want to try it out, then flashcard apps are pretty helpful. Even like Quizlet, I think, has like flashcards. I know some people that use that. Okay. Yeah, important as well, especially if you're in year 11 as well. This is your first 3, 4. Getting into that idea of having an exam at the end of the year as opposed to in the middle and at the end. And so with that, I mean, I'll get into this stuff in Q&A, but it's about constant revision. The stuff that I'll talk about, like things that you learn in Unit 3, Area of Study 1, don't do the SAC in like March or April, whenever, oh, probably March, and then never touch it again until September, October. You need to be constantly revising it. So then by the time you do get into exam prep, you're not having to relearn stuff. It's already kind of at the front of your mind because you've been referring to it the whole year. Okay, really, really good, I guess, concerns. They are not good concerns, but very valid concerns. And obviously, hopefully things that we'll address during the lecture. But yeah, a lot of the stuff is content, and we'll get to this kind of stuff in Q&A, but about how to study the best and kind of manage the content and work through it and stuff like that as well. Okay, to not waste any more time, let's jump into it. So we're looking at Area of Study 1, which is very much DNA stuff, really. So we're kind of thinking really little minute details of DNA and stuff like that. Okay, so essentially, most of you would be familiar with this, I'm assuming, but this idea of what DNA is, what nucleic acids are. So we're thinking of DNA and RNA. So essentially these 3 things, understand these because there have been questions where you have to label them. So we have our phosphate group, we've got our sugar, and then we've got our nitrogenous base. So we've got our AGCT in our RNA. We've also got our uracil as well. And you can see the study design dot point that covers this. So in terms of RNA and DNA, the main difference is, again, the type of sugar. So DNA, deoxyribose sugar, RNA, ribose. Be aware of this. You may get questions that will trip you up. I think I remember it might have been like 2018, something like that. It was like label, like a diagram like this of RNA. And I think so many people would have just said sugar, or so many people would have written deoxyribose sugar, just assuming, because I didn't read it properly. And the only accepted answer would have been ribose sugar. So you have to be specific and you have to understand when you're talking about which nucleic acid. So we can see deoxyribonucleic acid is double-stranded and ribonucleic acid is, RNA is single-stranded and tends to be a little bit shorter as well. Okay, so in terms of polymers, I'm not going to go over this stuff too much. It's not as vital in the study design, I would say, but the thing to understand is this idea of hydrogen bonds. So our little bases, we can see that this is our monomer. So heaps of them together make up our polymer, right? Like mono, poly. So when we have our kind of long nucleic acid, you can see we get this little ladder. So each of our bases kind of connect to a base, on the other side of the ladder, right? And via hydrogen bonds, that's how they connect. If you do chem, you'll probably be familiar with this. If you don't, again, you're not at a disadvantage, but you just need to understand what you need to know. So you can see C goes with G, A goes with T, and understand that they're connected via hydrogen bonds. So when you talk about DNA and stuff, if you talk about it at this level, that's what you should be referring to. The rest of the stuff isn't too important. Again, understand that it's anti-parallel as well. Okay, this is the important stuff. So you can see DNA and RNA, and you have to know what's different. So uracil versus thymine. Again, understand this stuff. It's good when you can compose this kind of thing in a diagram. So you can see a table here. If you can pop this stuff, I mean, again, it depends on the type of learner that you are, but you can like basically contain all this information in a diagram as well. In bio, diagrams are super, super, super helpful for memorization, especially because a lot of the stuff is to do with processes. So like, your flow charts, your diagrams, even more mind maps in terms of concept relation, that can be helpful as well. So yeah, we're thinking double-stranded versus single-stranded. thymine and uracil, and the sugar, obviously. And their roles are obviously different as well, which we'll get into. So really important to understand the 3 RNA types. So you have your mRNA, your rRNA, and your tRNA. So mRNA is the main one that we'll talk about because it's very involved, obviously, with protein production. So mRNA, basically, the idea is that it is the, I guess, basis of kind of forming that protein, or gives the specific instructions to form a certain protein. RNA is like the one you least look at. It's just a structural component of ribosomes. That's all you need to know. You don't really talk about anything else to do with that. And then tRNA, that's important as well, involved very much in our, again, protein productions, our translation. And yeah, it basically just brings your amino acids during that process. But we'll talk about that. Essentially, yeah, you just have to understand when they are separate, and again, be specific as well. If we are talking about RNA, you're talking about mRNA, you're talking about tRNA, and then just understand that rRNA is part of a ribosome, really. Okay, in terms of gene expression, so with bio, I want you to try, especially with stuff like this where it's so minute, like where you're thinking of like little mechanisms inside a cell, it's very good when you can, like, to understand it, look at it, like take a step back and look at it from a bigger picture. So understand gene expression. Don't just memorize, transcription and translation is part of gene expression. Think like, what is gene expression? We're trying to, we've got our DNA, which is in our little nucleus, giving us the instructions for everything we need to do. And how we get that information and take it into a protein that actually performs that action, if that sort of makes sense. So understand which words are important. So we've got our gene expression, the idea of the genetic code being degenerate or redundant as well. I will say definitions are really important in bio. Start making a glossary. If you didn't really do that in 1-2, do it in 3-4. Having a glossary and adding to it and constantly revising it will help you a lot, especially with short answer questions that are definition based. Okay, so gene, basically it's the sequence of DNA that codes for that certain protein. Gene expression, we're thinking about the steps of transcription and translation. So again, if you see the term gene expression, don't think about like, oh, what is this complex thing? Like, it's literally just think of it, break it down, transcription and our translation. So getting our gene and creating, our little mRNA, basically our code for our polypeptide, and then producing that actual polypeptide or that actual protein in translation. And then the idea of referring to the, here, as you can see, universal triplet code that is degenerate. So degenerate just means that you can have multiple triplets. I mean, triplet, I would prefer to say like codons. You can have multiple codons that will code for the same amino acid. So you can have one amino acid and 4 different codons can produce it. So it's this idea that you can have different actual bases, but the same end product, if that sort of makes sense. Yeah, so understand that as well. Okay, so obviously you can see the steps of transcription and translation are fairly complex. As we go through this lecture, you'll see a lot of slides like this, when you go into photosynthesis, cellular respiration of complex processes. Again, we're covering a lot of content. We're doing it in a short amount of time, so I will go through it as like pretty briefly. If you've got really like a lot more specific questions or you didn't catch something that I say, then pop it in the Q&A. But understand, you will get these slides as well. Obviously, you're going to be taught it in a lot more depth. So if you're not grasping every single detail immediately, don't think like, oh my gosh, I'm so silly, like I want to fall behind. Like, understand, we're just giving a brief overview and we're going through the general steps. So don't feel too concerned. Yeah. Okay, so basically we've got our 3 steps of transcription. So you have your RNA polymerase. So this is a really important enzyme to be aware of. And that binds to the promoter of the template strand. Stuff like template and complementary strand, once you revise it enough, you'll get it. But basically you think we have our RNA polymerase. So think about what it's going to do. Ase means it's an enzyme. Polymerase, it's going to make our polymer of RNA. So the DNA will basically unwind, as you can see here. So obviously it's double-stranded. It's a nice tight coil. To get, remember, we just need one strand. We're just going to copy one strand. So we have to unzip that DNA, and our RNA polymerase, I don't think it's drawn here, but our RNA polymerase will basically, oh no, this whole thing is RNA polymerase. Okay, we'll move along and start essentially synthesizing that RNA. So we can see that it unzips it, and it will do this by complementary base pairing. So remember, our A goes to our T, our C goes to our G. I know there's like a lot of memory aids to kind of remember that. I think there's like apple in a tree, car in a garage. I just used to remember it because C and G like look the same. Like they're both curvy. So that's how I remembered it. But very important to just have those pairs in your head. So anyway, it goes through and pairs those using complementary base pairing. Remember, we're thinking of hydrogen bonds attaching them. And you can see that this, as RNA polymerase moves through the gene, essentially unzipping that and going through, we're getting this strand that's created. And then the DNA just winds back up as the RNA polymerase moves through. Ultimately, we get to the end of the gene. We've got our nice long RNA strand created. We'll reach this termination sequence, and that will tell the RNA polymerase, okay, get off the gene now, like you can detach, we're finished. So basically the RNA polymerase will hop off, and that little RNA strand, which is called pre-mRNA, that will be released, and it'll go into our modifications ultimately. So understand that in our little DNA strand here, this is the coding strand, this is the template strand. Remember, they're complementary, right? Because usually they're just sitting in their little coil. Our RNA is also complementary to the template strand. Therefore, it is exactly the same sequence as the coding strand, but instead of T, it's got a U because it's RNA. Really, really remember this point. You see it in multiple choice questions all the time. And it's alluding to the fact that thymine is in our DNA, uracil is in our RNA. So just always understand it. You'll come across so many questions where they try to trick you with that. Okay, so that's our first step. So we've gotten our DNA and we've used that to create our mRNA or our pre-mRNA. So to allow this pre-mRNA to basically go into the cell and to enter translation and to go with the ribosome and stuff like that, we have to modify it a little bit so that it can actually leave the nucleus. Remember that our DNA is in our nucleus, this is all happening there. We want to send our mRNA strand into the cytosol, the cytoplasm essentially, so we have to prepare it a little bit. So we've got three main things that go on. So our introns are removed. So basically in DNA, we've got little sections that don't code for anything. So they're essentially a little bit worthless when we want to produce our protein. So we get rid of them. That is why mRNA is a little bit shorter. We also have our little cap. So think, something on our head, and then I just think like something on our teeth. Oh, sorry, not our teeth, our feet. Or I guess like our backs, if it's a tail. So just think a cap and a tail. It makes sense. One at the top, one at the bottom. So you have your methylguanosine cap or your 5' cap. You hear it referred to as a lot of different names. But basically your methyl cap, that's added to the 5' end, and your poly A tail, which is just a basically long strand of adenines, really. They're added to the end. You don't need to know too much about these, just you have to understand that these are added before translation, essentially. So you've got, your methyl cap, your poly A tail. Now that these have been done, you've got your intron spliced out, we've got our mRNA molecule. That's when you can refer to it as mature mRNA as mRNA. Technically, I think there is a bit of leniency, like if you are to refer to something as mRNA before this step, but if you do want to be specific, and I don't know, some teachers might be really picky as well, you should refer to it as mRNA only after this step, and before that, it should be called pre-mRNA. So hopefully that is making sense as well. Okay, now think of this little mRNA strand moving out of the nucleus and into the cytosol or the cytoplasm. And this is where translation occurs. Do I have a diagram? Yeah, we'll look at a diagram after. So it's out of the nucleus, it's going to a ribosome. So it enters the ribosome, it attaches. We have our start codon. So this is AUG, and this codes for methionine. I know it sounds like, but you'll write about it so often that you'll begin to remember it. But it's always the same. This is always the first amino acid. It's always the first start codon, and it tells the ribosome, like, okay, translation is about to start. We're going to produce our polypeptide now. So we have our start coding. tRNA will bring our little methionine, and then we'll start to attach our ribosomes. Our amino acids. It'll make more sense when I look at the diagram. But basically, we've got our AUG. We will then move on to the next codon. So remember, we've got our mRNA. It's made up of heaps of A, U's, G's, and C's. We've got all our letters, and we read them by threes. So we read them by codons. And so each codon, again, as I mentioned, will code for a specific amino acid. So we read AUG. The next one, maybe it's AAA. The next one, maybe it's CUA. And they'll code for specific amino acids. And the tRNA will have the complementary codon and will bring that. So then we'll get our next amino acid and our next amino acid and our next amino acid. And then we'll be forming this polypeptide chain. So you can see that in step 3. So again, it goes on and on and on. The ribosome basically moves along that mRNA strand, keeps going, keeps attaching those polypeptides until eventually we reach the stop codon. So the stop codon, that'll come at the end of the mRNA strand, basically. This does not code for anything. Please, please, please remember this. I think it's easy to get confused because the start codon codes for an amino acid, codes for methionine. The stop codon does not code for anything. Once the ribosome gets to the stop codon, translation finishes, the ribosome detaches, the polypeptide chain is released as well. So really remember that, because a lot of people get confused and think that a stop codon will code for an amino acid as well. It doesn't. Okay, so here is the diagram. So this is our little ribosome, and this is our mRNA molecule. And so this is what I mean by our little codon. So these are our codons as part of the mRNA. This is part of the tRNA. Can you see? So this whole thing here is tRNA. So you can see these little squares are the amino acids, and the thing at the end is the anticodon. So again, complementary base pairing. With our nucleic acids, it's all about this complementary base pairing. So you can see we have our AUC. The complementary of that is UAG. Remember, it's RNA. So instead of A and T, it's A and U. So our A, our complementary is our U. Our complementary is our A. Our complementary is our G. You can see here, GCC, the complementary of that is CGG. So that is our anticodon, and that's part of our tRNA. And so tRNA has an anticodon that matches, this certain amino acid, and so it'll bring them. So the next one will be GCU, and so on. And so you can see that they'll bring their little amino acids, and these will connect, and they'll form a polypeptide chain, which is basically our the basic version of our protein. And then so eventually we'll reach a stop codon. We will not get amino acid added when we reach our stop codon. This just signals like translation is over and everything detaches. And then our polypeptide chain is released. Hopefully that makes sense. Again, be aware, transcription, translation, they're very complex processes. I've talked to them fairly quickly. It'll take a minute to sink in. And again, once you're taught it in more depth, you'll get the hang of it. It does take a couple of times to really get the process in your head. Okay, so it's saying that. We'll go to a Slido poll now. Okay. So you'll get so many questions like this. Honestly, they can be a little bit tricky. I feel like this is a bit of a mean one to put, especially if that's like the first time you've heard about translation. But just give it your best. Again, I'm not expecting everyone to get it right like the first time, so don't worry. So if an mRNA strand is made up of 90 bases, so we have 90 bases in our mRNA strand, yeah? So thinking of after transcription, how many amino acids will our polypeptide chain be? What do you guys think? So our mRNA strand, again, think about codons and that. Okay, I won't give you guys too long for all of our Slido polls because again, I just want to be mindful of time. So the answer here is 29. So, again, I'm sure this might be for most of you. I'm assuming this is probably like the first question that you've seen like this. Again, throughout the year, you'll get so used to questions like this, you'll get a billion of them. And I guess it's like a little bit of maths, but not too much. So you have 90 bases. So obviously, you divide them by 3 because of the codons. So then that will give you, you'd think like 30 codons. However, remember that there is a stop codon. The stop codon does not code for an amino acid. So that's why it's 29. So you've got 30 codons. All of them will code for an amino acid except for the last stop codon. So that's why you minus 1. So that's 29. Yeah, you'll get this same question a billion times with obviously a different number here. You just have to basically divide it by 3 and then minus 1 for the stop codon. Okay. So moving on. Now we've gotten our little polypeptide chain. Again, pay attention to the study design and what's written here. So we've got our polypeptide chain. We finished translation. So we've, this little thing is completed. To make it into a functional protein, we have to fold it. So again, this kind of gets a bit like you cover this sort of stuff in chemistry as well. This is where it kind of gets a bit scientific-y. Obviously bio is a science, but like we're getting into stuff with bonds and things like that. So the bonds between our amino acids are very strong. They're not like the bonds between our nitrogenous bases that were our hydrogen bonds. Those are a little bit weak, and that's why we can break, like, when our DNA unzips for our transcription, like that's a fairly easy thing to do because the hydrogen bonds aren't that strong. However, the bonds between our amino acids, I think we say it here, it's like peptide bonding. So these are, again, if you do chemistry, covalent bonds. They're very strong. So basically the polypeptide chain is pretty like locked in where it is. However, when we fold it, these are often like our hydrogen bonds. We've got other things here. These are all a little bit weaker. They're just kind of semi connections. I don't want to, again, get into too much like chem talk. But basically we've got 4 levels. We have our primary, our secondary, our tertiary, quaternary, and each time it's just a new level of folding, And by the end, we have our functional protein. So this is our most basic level. This is our primary. This is literally just what it is at the end of translation. So we've got our specific amino acid sequence. Literally, it's just this, what the actual amino acids are. When we have our secondary, this is when we get our kind of initial folding. These are our hydrogen bonds. The important things to be aware of are these names here. An alpha helix or alpha helices, and our beta-plated sheets. So you can see them here. It's not too big to kind of know, like, especially like the intimate details of them and like generally have an idea of what they look like. But basically our alpha helix, they're just like helices, like little coils. Our beta-plated sheets, just these flat sheets. And so this is kind of our initial levels of folding of our protein. So we're getting, essentially the bonds are occurring between the amino acids. Again, I don't want to get too specific. You guys will learn about this, but amino acids, like obviously they all look like the same little circles here, but at like a molecular level, they've got different groups and different elements and stuff like that. So that's why you get different folding based on what amino acids you have. So, methionine may form an alpha helix with, I don't know, whatever other, like cysteine or something like that. It just depends on the amino acids. And so that's why with all of our different proteins, they're all folded differently. It's very unique because they all have a different primary kind of basic level of folding. They're all a different sequence of amino acids. Again, hopefully this is kind of making sense. So yeah, we've got our little string of amino acids. So like, I don't know, you can literally think of like a little necklace, a little beaded necklace. And then we've got our initial folding where some of these little beads on the necklace like other beads. So they want to come closer to them. Like you can kind of think of that. So we've got a bit of initial folding, and this is forming our alpha helix, our beta plated sheet. And then we're moving on to our tertiary level. And this is where the main stuff is. This is where we get the main structure of the protein. So we're folding a little bit more, and these are a bit more complex than our little simple hydrogen bonds. So we're getting ionic stuff, we're getting disulfide bridges, basically. It's just this like two sulfide stuff. We can get like hydrophobic stuff. We can get hydrogen bonds as well. You'll learn more about it, but basically this is a lot more folding. So with your initial, little pearl necklace, one pearl wanted to head over here. It's a little bit basic, right? Whereas here, it becomes so much more complex. Like now the whole thing is kind of contorting itself because you've got a lot more bonds going on. And again, this is the same principle. It's because all of these little individual amino acids have got little different structures and different groups, and they like to form bonds with the groups on other amino acids. So you get this really wacky, folding and shape going on. And this is the tertiary structure. And so that's what we've got going on there. And this will actually, like, a protein can function at its tertiary level. I know we've got quaternary here. You can see that it's optional. Some proteins will just stick to tertiary. And this is what gives it its shape and its role. One thing I really, really, really want to emphasize is with proteins, the structure equals function. The shape that we've folded, that will lead the protein to be able to perform its function, basically. So it's really, really important. If anything goes wrong with the folding, if anything in a protein is affected in terms of its structure, it will affect its function. A protein cannot perform its function if its structure is out of whack. Okay, and then lastly, we've got our quaternary kind of level of folding. So this is when you basically have multiple polypeptide chains. So they're all separate. They've all got their own little tertiary structures, and then they come together to form basically this other bigger protein. So you can see here, I think this is hemoglobin. So that kind of protein that's found like within blood. So there's 4. So this is quaternary folding, and it's basically when 4 separate polypeptides, they come together. And again, it's just different bonds and different attachments. And then they create a bigger functional protein as well. Again, it's relatively complex. You'll learn more about it and it'll make more sense, but that's just an overview. Okay, so you have your genome and your proteome. Again, these are definition things. Chuck them in your glossary. Basically, genome is the kind of summarization of all the genes in the human body. The proteome is the same thing, except the protein version. So it's basically all the proteins that are in a cell in the organism, everything like that. And it is really important. Essentially, you can see like the collection of proteins that will basically tell you what the organism does, if that sort of makes sense. So the genome is like the basis, like the instructions, but the proteome is actually like what's going on, what's there. So you can get a better insight into kind of what's going on in a cell, what's going on in a human, when you look at the proteome in terms of like what the actual proteins being expressed are, as opposed to the genome. It's kind of like a genome is like looking at, I don't know, all the ingredients you have separately, whereas the proteome is like looking at the meals that have been made from those ingredients. Hopefully that kind of makes sense. Honestly, you don't get too many questions on this. Basically, just know their definitions. Okay, so another thing we're looking at is gene regulation. Again, take a step back. Before we zoom in and look at all of this stuff, understand what it says on the study design. Prokaryotic gene regulation, so operons. We're not thinking about our cells. We're not thinking about eukaryotes. We're thinking about very simple prokaryotes, so such as like bacteria, stuff like that. So in terms of our operon, our thing with our operon is basically it's like it chucks a whole bunch of genes together and treats them as one gene. So it's almost like, in this case, like 6, like 5 in 1. So you can see here are the structural genes that we have all these different genes, but they're clustered together and they will only be basically transcribed as one. So rather than like transcription, for this gene and then that gene and then that gene, these genes all kind of work together for the same purpose. So the prokaryote just goes, okay, I'm going to chuck them in an operon. And when I need them, like I'll need them all at the same time. So then I'll just transcribe the whole operon. Basically, that's what we're looking at there. I don't know if there's more. Okay, so what we will do is you can see, I know this system again looks complex, but basically the prokaryote has a way of regulating when to produce these genes and when to transcribe them. So the one we are looking at here is the trip operon. So it's for producing tryptophan. And so tryptophan is basically just an amino acid. Like it will, obviously have a role with protein production and all that kind of stuff. So the whole system with this is these genes will lead to the production of tryptophan. So we will need to transcribe them when we are low on tryptophan, right? So think of our little bacteria hopping along. The bacteria has got a low level of tryptophan. We're going to want to transcribe these genes, right? We're going to want to produce some more. On the flip side, let's say we've got so much tryptophan in the cell, we have heaps. We're going to want to turn this off. We're going to want to shut it down, right? So the prokaryote has got this very smart system of regulating it by using the tryptophan itself. So we have a little repressor. So what a repressor does is basically it will just sit on this region known as the operator, and it can block transcription, basically. So again, I'm going to try to talk through this simply without confusing you too much. So we have our little repressor. Let us say that we have a lot of tryptophan in the cell. So think we've got this little bacteria. There's heaps of tryptophan. We don't want to produce any more. So we want to turn this operon off, right? It'll be a waste of our time, a waste of our resources to keep on producing tryptophan when we've got too much of it in the cell. So what happens is the tryptophan here, it will bind to this repressor and basically turn it on. It will activate it. So you can see that here's the repressor without tryptophan. It looks different to when it has tryptophan bound to it. So tryptophan attaches, it is now in its active form. The repressor wants to repress. It will sit on the operator. And remember, RNA polymerase binds here to the promoter. RNA polymerase will not be able to get through. It cannot transcribe that gene. We're not going to get more tryptophan. That's the basic gist of it. So you can think on the flip side, if we've got a low level of this amino acid, we've got a low level of tryptophan, our repressor is basically in its inactive form. It cannot attach. So then RNA polymerase will, be free to transcribe that basically. Hopefully that makes sense. Again, it's a process. Diagrams help a lot. You'll talk through it. So if it's not clicking immediately, don't worry, but just understand it's this really, amazing idea of how to regulate the level of tryptophan in the cell. The prokaryote uses the actual molecule itself. So it's quite, it's very logical. It makes sense. You know what I mean? Like when you don't have enough tryptophan, the thing that will block the production of tryptophan literally can't. When you have too much tryptophan, the tryptophan will be the actual thing that blocks its own production. So it's almost like, I don't want to say self-regulation, but you can see how the tryptophan is very involved in its own regulation process. So I hope that makes sense. There is another process called attenuation. I'm not going to go into it in too much detail because it is a little bit more complex. And again, you'll learn about it and stuff like that. But basically, there is a leader section that you can see here. So you see how this is separate to our structural genes. It doesn't really have any involvement with the actual production of tryptophan itself, but there's a second form of regulation. So we've got this, our little repressor. This leader section is involved with this thing of attenuation. So what it means is within this little gene, to produce the protein from this, so to kind of, transcribe it for mRNA and then translate it, you need tryptophan. So you need tryptophan. So what happens is when there is a low level of tryptophan, the ribosome basically moves along it a little bit slowly because it basically has to wait for the cell to kind of bring it some tryptophan, right? Because there's a low level. We don't have a lot floating around. So the ribosome basically just takes its time and waits because it needs more. And what that does is it means that RNA polymerase basically just continues to go through and transcribe this operon so that we're producing more tryptophan, which is what we want, remember, because we have a low level. On the other hand, if we've got a high level of tryptophan in the cell, it's floating around everywhere, there's way too much of it, the ribosome basically can transcribe really quickly. And then so it, again, there's more technical stuff, like it has basically this like hairpin formation. Again, I'm not going to go into it too much, you will learn about it. But it moves so quickly and ultimately the ribosome basically falls off. And then so RNA polymerase just stops. And that's why you get this attenuated mRNA and why that's really short. And again, that makes sense because we have such a high level of tryptophan, there's no reason to make more. So hopefully that kind of makes sense. I think attenuation is a little bit of a tricky process. It does take a while for it to sink in. The main thing of this is obviously gene regulation, but again, step back, think, why does Vika want me to learn about this? A big part of this as well is to understand, like with attenuation, it can happen because in prokaryotes, transcription and translation can happen at the same time. Think about it in eukaryotes. Transcription happens in the nucleus, translation happens in the ribosome, in the cytoplasm, in the cytosol. You cannot have those two processes at the same time. Transcription has to be done for translation to start. So that's something to be aware of. Again, this is probably like one of the most complex things in this protein stuff. I'm going to skip the VCAR questions. Feel free to look at them in your own time. It's just because we've got a lot of content to get through. Okay, that's it for protein basics. I know this says basics, but it's very complex. Transcription, translation, even the operon stuff, it takes a lot and it's complex. It's long processes. So revisit it a few times. Your teacher will teach it to you as well. Look at different resources. And after a while, it'll just sit in and it'll click. Okay, now we're looking at DNA manipulation. So those are our kind of basics of protein, how we create protein in the cell, how DNA kind of works. Now we're thinking about manipulating this process. So we have DNA polymerase, DNA ligase, and endonuclease. So these are our important enzymes. Don't really worry about helicase too much. So DNA polymerase, you don't look at the intricate details of actually synthesizing DNA, so don't worry about that at all. You just have to know what DNA polymerase does. Think of the name, DNA polymerase. What does RNA polymerase do? It makes an RNA strand. DNA polymerase, it makes new DNA, basically. DNA ligase, think of this as like ligating something. It's basically glue. So it actually joins the backbone. So if you remember our little diagram, we had our phosphate and our sugar. Ligase attaches those. Those are very strong bonds. Remember how we talked about our little hydrogen bonds? So between our little nitrogenous bases, these are hydrogen bonds, pretty weak. That's why we can separate them, unzip them, all that sort of stuff. Between our actual, like our sugar phosphate backbone, so what's highlighted here in green, these are very strong bonds. And that's why we need a DNA ligase. We need an actual enzyme to help us because they're very strong. Don't worry about Okazaki fragments or anything like that. It's beyond the level. Endonuclease, this is really important when we talk about DNA manipulation. Just our little scissors. So if DNA ligase is our glue or like our sticky tape, endonuclease are our scissors. So this is what cuts DNA. Okay, so going into this a little bit more, basically they can cut them with different ends. So you can cut them straight down the middle and this will produce blunt ends, or we can cut them a little bit jagged, I guess, and then we get sticky ends. So in terms of why you would have different ones, it just depends on the endonuclease. So here are our lists of endonucleases. You may also see them as restriction enzymes. They're just the exact same thing. And they have their own recognition sequences. So this is like they're literally programmed. If they see DNA like this, then that's where they'll cut it. That's where they'll cut it. So you can see if it's down the middle. So like if you've got 2 on the left and 2 on the right, then it's going to be this blunt end. If you've got, it's like cutting kind of 1 on the left, whatever that is, like 5 on the right, it's going to produce this sticky end. So in general, when we think of DNA manipulation, our sticky ends are a little bit better because we've basically got little like tags, I want to say. So think about our complementary base pairing. AA and TT, they're only going to pair with TT and AA, right? So if you were to chop the DNA up, you've got two ends like this. Those will reform very specifically. AA and TT, will not reform with another sticky end that is CGAT, right? Because it's not complementary. It'll only pair with TT and AA. So therefore, when you're manipulating DNA, sticky ends are often a little bit more favorable because you know that they'll be like sticking together properly. Whereas blunt ends, because there's no like overhang, they can attach to anything. So like, say we've got this, so, you know, GG and CC, these blunt ends, they can stick with anything. Doesn't matter what's on the left. It could be, you know, CC and GG, or it could be A, T, CC, it could be CU, you know, whatever, not U. But yeah, so that's why blunt ends can be like, I guess a little bit worse, but that's just something to be aware of. Okay, so CRISPR-Cas9, this is kind of a complex system that we look at. So it's a naturally occurring thing that happens in bacteria, and basically we can use this system to manipulate DNA. So CRISPR-Cas9, this will make a lot more sense, or I think it helps a lot when you learn it after you've learned about immunity, like human immunity, because basically this is like a bacteria version. So in general, how CRISPR works is you have a little bacteria, a virus attacks it, obviously like, oh, that sucks, right? So what the bacteria will do is, okay, they're going to form their own sort of like immune system. So they will basically take the virus's RNA or DNA. So you can see to attack a bacteria, these are our little bacteriophages, so they're viruses. They insert their genetic material into the bacteria, and then obviously it creates more viruses, and that's not good. So the bacteria takes a little bit of this genetic material and it stores it within its own genome. So it's like, you know, it just takes like a little bit of, it's almost like taking a photo, I guess you could say. So like it takes a little photo of this virus and it stores it in its pocket so that the next time this virus comes along, the bacteria will be able to pull it out of its pocket, compare it, and then kind of mount a response really quickly. That's how it uses CRISPR-Cas9. So we take our little photo, our little snapshot of some of this DNA, and we basically stick it into the bacterial genome. So when this virus comes again and attacks the cell, the little, I guess, genetic material, which is stored in our CRISPR sequence. So our CRISPR sequence is basically like our little, if we're thinking of these little viral things as photos, CRISPR is like our photo album. It's where we're storing these photos of our different viruses. So we have our CRISPR sequence. Basically, our little virus comes along again. What the cell does is, because, you know, we've got our photo album, we basically take out this little photo, we transcribe that genetic material. Yeah, we transcribe this genetic material, and then we use Cas9. So Cas9 is an endonuclease. So remember what we've just talked about, our scissors. Its job is just simply to cut. So we have our little genetic material. Can you kind of see where this is going? We've got our genetic material that matches the viruses. We've got our Cas9, our little scissors. What this system does is it goes to this new virus that's inserting its DNA. Cas9, our endonuclease, recognizes it, cuts it. The virus cannot replicate. And that's how it's like a bit of an immune system, if that sort of makes sense. So yeah, Cas9 basically detects that because the DNA is complementary, and then it just cuts that. And so you can see the virus can't attack the cell anymore. So that's how it works naturally in bacteria. What we've done is kind of looked at this and used it for DNA manipulation, because basically if you can program Cas9, instead of looking at, you know, a gene sequence that matches viral DNA or RNA, it can match perhaps like a gene that causes cancer or something like that, and then Cas9 can cut it, and then now that gene is inactivated. So you can see how that's a little bit of DNA manipulation, essentially. Hopefully this might make more sense. So this is our enzyme. This is our guide RNA. So you can see when the Cas9 basically meets RNA that's complementary, it cuts it. And so in terms of talking about DNA manipulation, you could insert a gene of interest. So like, I don't know, for example, like if you wanted, let's talk about it in the context of plants or something, like maybe this gene will make the plant grow really, really big, or the flowers will be really red or something like that. So you can cut it, that's Cas9's job, and then using our other little techniques, you can insert a gene into that little gap. Okay, so moving on to other stuff, we've got this idea of DNA profiling. There are 2 steps of it, and you have to know these processes as well. So PCR and gel electrophoresis. So I'm sure you'll be pretty familiar with what PCR is. In terms of its steps, it's not too complex, but you do have to have a couple of things to start off with. So the point of PCR, polymerase chain reaction, we are amplifying DNA. We're starting off with a little bit. So like, you know, talking about like our COVID tests, like we've gotten a little bit of like this viral DNA or whatever's, you know, in your little nose swab, and we're going to amplify it. So we have heaps of it, and then we're going to look and see if it's, you know, COVID DNA. Or like if you're at a crime scene and there's a drop of blood, you've got a tiny bit of DNA, use PCR to amplify it. So now you have heaps of it, and then you can analyze it and like find out who the criminal is, or whatever. Okay. So to do this, you need a couple of things. So DNA primers, so basically just little short bits of DNA, DNA nucleotides. So obviously if you're going to amplify DNA, if you're going to like synthesize DNA, I guess, you need the nucleotides, and Taq polymerase. So this is basically a specific DNA polymerase that allows you to do it in really hot conditions, because we'll have a look at the steps of PCR. Basically, you can see that it goes up to 95, 72. It's pretty hot. So you need to use a polymerase that can withstand that heat. So this specific one is what works. So, okay. Basically, we've got our initial, I don't know if we have we have a diagram here. This will make a lot more sense after I talk through the steps, and then you look at this diagram. But you can see you start off with 1, you can end up with like a billion strands. So firstly, the PCR mix. So what we've talked about, you heat this to 95 degrees and that denatures, it basically splits the double-stranded DNA into 2. Remember, it's our hydrogen bonds. They're not that strong. So if you just heat it nice and high, that is what splits it up. So you can see here, double-stranded, you heat it really nice and high, you split it into 2. Then we want our DNA primers to kind of come along. So you cool it to 55. If you were to keep the temperature really high, obviously you can't have complementary base pairing. The hydrogen bonds won't form. So you cool it a little bit back down, so 55 degrees, and this is where our primers, so think about what does a primer do? Basically start something off. It's like the foundation, right? So our primers come and they attach to each of these little strands, and that's what will kind of get the ball rolling. You then heat it back up to 72 degrees, and this is when Taq polymerase can just work really nice and fast. So you turn it up a little bit, and your Taq polymerase, remember, it's just a DNA polymerase. It will synthesize those DNA strands. So it will bind to the primers, it'll start off there, and then it will go on and just synthesize as normal. It'll bring the complementary bases and form a new DNA strand. So then you can see what we've got here. We've got now double the DNA. We started off with 1 DNA strand. Now we've got 2. So then we repeat that process again. So we denature them. Now we've got 4 strands. We attach the primers. We've got Taq polymerase. So now we've got 4 pieces of DNA. So we had 1 piece of DNA, then we had 2. Now we have 4. Next we'll have 8, so on, so on, so on. So after a couple of rounds, you can see that just increases exponentially, and then we get heaps of DNA. So understand that these are the 3 main steps. You have to mention this idea of repeating it. If you write in your short answer, 95, 55, 72, that's it. it implies that you only did one cycle. So if you had one piece of DNA to start off with, you had two at the end. Like, that's not what PCR is meant for. You want to amplify it, you want heaps. So you need to remember to write about repeating this cycle. Okay, the last thing is gel electrophoresis. So basically, this will determine the size of DNA, and it creates your DNA profile. So there's a lot of applications. Again, I'll just talk through the process generally. Like, you'll learn about applications and stuff like that. But it's basically so you can paint a picture of what the DNA kind of looks like. Because technically what you're getting is, this just shows what size DNA strands are. But if you think about using like human genes for this, like if you use my genes compared to, you know, my mum's or like my siblings, stuff like that, you'll all have different ones. And so that's how you can use this to kind of identify certain genes and things like that. So the basic principle is you have this little, it's like jelly. That's the kind of consistency of it. So you put these little, like you put the DNA in the wells at the start, and because DNA is slightly negative, remember this, DNA is slightly negative, so I think it's like the phosphate group, something like that, it will move towards the positive side. So electrophoresis, that's where the electro part comes in. So they start off at the negative side, and because, you know, negative attracts positive, they're going to migrate towards the positive electrode, and that's where we're going to get this separation. So think of this jelly basically like a sponge. There's little porous holes. So if you are a tiny short piece of DNA, you find it easy to just weave in through these holes and to wiggle through, and you get to the end really quickly. If you are a longer piece of DNA, you are going to have to, you know, you're going to move through these holes a little bit slower because there's more of you to get through. So therefore you're going to not travel as far. And that's what the separation is. So you have your longer bands towards the start, basically, and your shorter bands towards the end, because the shorter bands move faster. And then so there's a lot of applications for that, as you can imagine. So you can determine sort of length of DNA and stuff like that. Hopefully that makes sense. Okay, cytopol. Again, I know this is a lot of information being thrown at you. Just try to digest it as best as you can. Okay, ranking. So the correct order of the steps for PCR. So do you guys remember what the degrees that you need are? Okay, won't give you too long. But yeah, okay, looking good. So yeah, denaturation, annealing, elongation. So really hot. And remember, you have to know the purposes of these. It's easier for stuff like this, like and things with bio, and again, like literally all your subjects, try your hardest to not just rote memorize stuff. So think like, okay, 95, 55, 72, 95, 55, 72, and just try to remember it like that. Have a deeper understanding. Why do I have to warm it to 95? I have to get it really nice and hot so that you can break the hydrogen bonds and separate the double strands. Why do I have to lower it? So that their hydrogen bonds can reform when we're thinking of our primers attaching. Why do I have to raise it again? Think of, yes, Taq polymerase. It works really well under heat. This is a good temperature for it to work nice and fast. Like, think about that with the processes of PCR, and that's a lot better, and that'll aid your understanding and give you a much deeper understanding than just rote memorizing, okay, 95, 55, 72, and trying to remember it like that. Okay. I think, let's see if there's any more. Okay, so there's a little bit more left on DNA manipulation. I'll leave this. So it's the use of recombinant plasmids to transform bacterial cells. This is a bit of a complex process as well. Again, you'll get it. I know I'm saying this a lot, but like, have faith, you'll get it. After you look at it, a lot of times you'll get it. So in terms of plasmids, basically in a bacteria, we have a lot of chromosomes. They have one circular chromosome. So just one circular chromosome. They also have these other little tiny sections of DNA called plasmids, and then just like little small extra circles of DNA. So they're a circle. They're not strands, long strands like ours. They're a circle. An important characteristic of this is that bacteria can share plasmids, so they can take up plasmids and I guess like send them out as well. So we basically use these plasmids again as vectors to insert genes that we want to be inserted into bacteria, and then bacteria will produce these. So for example, with insulin, we insert the insulin gene into a plasmid using our little tools. We then put that plasmid into a bacteria. Remember, a really big thing about bacteria is that they replicate super quickly. So you put our little basically human insulin gene into this bacteria. This bacteria will divide and divide and divide and divide really quickly. And then you have a bunch of insulin producing bacteria. They're producing the insulin. You then harvest that insulin, purify it, and then you have human insulin. So that's the general process. This will probably help you. That's what it looks like. So I've explained the general process in terms of how this happens. So you have your plasmid. This is just a plasmid. It's just normal. You cut it with our endonuclease. Remember our little scissors? They just chop it up. We get our little open plasmid now. We then get the gene of interest. So this is our human insulin gene. You have to cut it with the same restriction enzyme. Think, why would I need to cut it with the same restriction enzyme? Why can't I cut it like it's just scissors? Remember, these scissors have very specific recognition sequences. You can almost think of it as like, it's not, like the scissors that you used in primary school and they had like zigzags or they had like waves, like little patterns like that. Think of that. You can't attach paper that you've cut with a little zigzag scissor to one that you've cut with a wave scissor. It doesn't match up. You have to cut zigzag scissor with a zigzag scissor so they can fit in nicely. Think of that. So you cut your human insulin gene with the same endonuclease. So then think of sticky ends. They'll attach really nicely. So basically you put them together. So you get your human insulin gene, you whack it in that plasmid, and now we've got our recombinant plasmid. So this is an important part to remember. Recombinant plasmid, because we've like recombined the genetic material basically. So something else that we do is we add antibiotic resistance into this gene as well. So we've got this nice plasmid, we've got insulin as part of it, and we've also got a gene for antibiotic resistance. So we've got that in our plasmid now. So we will then use our recombinant plasmid, and we need to get this into our bacteria if we're going to produce our insulin, right? So don't really worry about this too much, but basically you just force the bacteria to take up this plasmid, and then you will use that antibiotic resistant gene to determine which bacteria have taken it up and which haven't, because some bacteria just won't take up the plasmid. So that's why the antibiotic resistant gene is really important as well. And also sometimes, the plasmids may not take up the gene, stuff like that. So basically that's why the antibiotic resistance is needed. It's like a little tag to say, which bacteria have got the plasmid that contains the insulin gene in it. So we have the bacteria, we put our antibiotic. Obviously, ones that don't have the antibiotic resistance gene and therefore don't have the plasmid, they'll die. So goodbye. Whatever's left obviously has got that antibiotic resistant gene, and that's got our insulin, and therefore we will then grow those and we'll get our insulin from there. Hopefully that kind of makes sense. Okay, so now moving on to GMOs. So basically GMOs and TGOs, genetically modified organism versus a transgenic organism. Think about the names. So genetically modified means anything that has had its genome altered in any way. So they've had a gene removed, they've had maybe a gene like swapped around or something like that, or you've kind of added a gene from maybe like a different organism of the same type, like the same species. Think about it, something like that. Genetically altered in any way. In a transgenic organism, this is a specific type of GMO. So understand that transgenic organisms are also genetically modified organisms. Think of it like an umbrella, because a transgenic organism, think of transgenic, you've got a gene that's come from another species. So that's being put into another organism. And then so obviously, you're going to show that gene, whatever the gene programs for. You're going to express that protein, express that characteristic. So therefore, it's genetically altered. So it's a genetically modified organism. Something important to understand, which is in this last dot point, all transgenic organisms are genetically modified, right? Because their genes have been modified. However, genetically modified, if you are a GMO, a genetically modified organism, you're not necessarily a TGO. So if you have a plant and this plant has a gene that makes them susceptible to a disease, you take that gene out. It's genetically altered. It's a genetically modified organism. It is not a transgenic organism because it hasn't had a gene from another species inserted into it. Hopefully that makes sense. That is such a common thing that can strip students up. Just think of it like an umbrella. GMOs are the umbrella. TGOs are just a specific type of GMO that can fit into that category. Okay, in terms of cotton, so cotton is basically just an example of a TGO, I think. Yeah, so TGO, again, it's just like a certain characteristic. So this one here, a natural insecticide against caterpillars. So basically makes them not susceptible to these certain caterpillars. You can have other TGOs that might be for a certain aesthetic characteristic or, again, like growing bigger fruit or crop yield, stuff like that. It's important to think about ethical conditions as well. With the new study design, I would say there's an emphasis on ethics, especially with research methods. So with your area of study 3 stuff, bioethics is being pushed a lot more. It's not so specifically outlined with GMOs and TGOs as it has been in the past, but because there is such a big emphasis for kind of ethics in general with area of study 3, it's important to kind of think about this stuff as well. Okay, so you can think about things like this. So ethical stuff, obviously, social considerations, biological considerations. So think about if you've got TGOs, if those were to, reproduce with other non-genetically modified things, then you've kind of mixed stuff up. Thinking about artificial selection, think about, future generations of that species, stuff like that, how they might be affected. Sometimes it's this idea of should humans be meddling around? Socially, think about economics. So if you've got a TGO and it's producing, really big fruit and whatever stuff like that, people are all going to want that. So it's kind of leaving other people in the dust. Other things like that as well. Take them into consideration. Okay, so now looking at area of study 2. So with enzymes, basically something that you need to know, again, I know I've said it so many times, but this sort of bigger picture stuff. Photosynthesis and cellular respiration, all about enzymes. All about enzymes. So everything that you learn about enzymes, you will apply it to both of these. The idea with a biochemical pathway is that each step of this pathway is basically mediated by enzymes. So anything that affects an enzyme will affect the biochemical pathway. So will affect photosynthesis, will affect cellular respiration. So tie these in together in your head. It's really, really important. With enzymes, this is a super important definition you'll have to know. Chuck it in your glossary, like have it as the first one. Biological catalyst. So it is a protein. Everything that we've spoken about with proteins applies to enzymes. Everything we speak about with enzymes applies to photosynthesis and cellular respiration. See how everything is really, really interlinked. So it's very important that you have a good idea of how concepts relate. So basically what they do is they lower the activation energy. So again, kind of getting into chem stuff here. It basically just speeds up the rate of a reaction. So extremely specific. Really, you can, again, I know I'm referring to a lot of stuff we've talked about before, but you can see with the protein stuff, we were talking about complementary base pairing. It's all about specificity and being complementary. This idea that things kind of fit each other perfectly. Coenzymes as well. So basically these are just small little molecules. So you can think about like ATP, your NADPH. We'll talk about this as well when we get into the actual chemical pathways. And they just assist the functioning and they're just important in some of the reactions as well. Okay, so photosynthesis and cellular respiration have these general equations and you need to know these. The word equation is really important. I would just suggest to know the chemical equation as well. With the one on the top, photosynthesis, you can know this one. So it's the uncancelled one. So water on the left, water on the right. For me, I always just stuck with the cancelled one, so the one that's a bit more simplified. So basically just scratch the water off here and make this 6. So 6 carbon dioxide, 6 water molecules, 1 glucose molecule, 6 oxygen molecules. Because yeah, it just balances it out. Technically though, you have 12 in, 6 out, but either is accepted. VCAR is pretty happy with either. Cellular respiration, literally just reversed. So you have your glucose, your oxygen, carbon dioxide and water. Sorry, this is old. Ignore that number. The table that we'll get to at the end, that is the one that's accepted now. So with this study design, the number basically changed. So with the old study design, it was 3638. Now it is 3032. So please do not talk about 36 or 38. Sorry about that. Just get rid of that. Stick to the table that you'll see in a second. Okay, this is a bit chemistry-ish, but basically you just have to know that the activation energy is lowered. So look at that in your own time if you are interested. In terms of enzyme activity, what affects it? I do want to talk about the idea of it being a protein again. So remember that structure equals function. So important with enzymes because you have your active site and that will bind with the actual substrate. So when I refer to substrate, it's essentially like the reactant. So it's basically what's going to become the products. So it's really important that they are complementary. And so for the substrate to fit into the active site, the structure of the protein has to obviously be correct. So in terms of things that affect it, we've got temperature, pH, substrate concentration, and certain forms of inhibition. If this affects a protein or affects an enzyme, then it's going to affect the rates of photosynthesis and it's going to affect the rates of cellular respiration as well. Remember, everything ties together. So temperature, basically you have to understand that as temperature increases, the rate will increase until you get to a certain optimum temperature. Then it plummets downhill really quickly because proteins will denature. So basically the folding, if it gets too hot, that folding basically gets destroyed. Those bonds are ruined really. And remember, what happens if we denature our protein? The folding is broken. What does that mean? It means the shape changes. What's the key thing with proteins? Structure equals function. And so therefore, if the shape is different, if the structure is different, the function is basically gone. So you can see how it's like a bit of a relatively slow increase with the temperature. As soon as you get past this point, it's a much steeper plummet because ultimately the higher and higher you heat that, the worse the structure is going to get damaged and then it can't perform its action anymore. So pH, basically kind of the same thing, optimal conditions. If you so these are the different enzymes. So obviously different enzymes have different optimum temperatures, optimum pHs. So it just depends. And so that's why you don't really want to vary too much out of those conditions. I do want to say with these as well, understand that the rate of reaction is really low, obviously. Technically, the rate of reaction does not stop. An enzyme just speeds it up. It doesn't mean, an enzyme is not used up in the reaction. So the reactants will still react to make the products, but it just happens so slow that the level is basically negligible. You can't really see it. So think about in our body, if we weren't able to use our enzymes, if we didn't have enzymes, the reactions would still happen, but extremely, extremely slow. So that's why if you've got an issue with the enzymes, your body can't function because it's so slow. It's like billions, gazillions of years, whatever it is. So that's why enzymes are really vital. Okay, in terms of substrate concentration, basically the more substrate you add, the faster the reaction can happen, right? Because you think about our little enzymes. Say we've got 6 enzymes. If you have 2 substrates, you've got that reaction happening. If you have all of those enzymes filled up, then you've got 6 substrates and that reaction is going to happen faster. However, it gets to a certain point and then it plateaus. Because if you have 6 enzymes, obviously as you add some substrate one by one, you're going to get this increase. It's going to go faster and faster and faster. But if you add 7, then you add 8, then you add 9, then you add 10 up to 500, you've still only got 6 enzymes, and they can only work as fast as they can work. They can't, like, basically the substrates just have to wait in line. So that's why you get a plateau. And the same thing happens if you increase enzyme concentration. If you have 10 substrates waiting and you have 1 enzyme, then you've got 2 enzymes, then you've got 3, you're increasing until, you get 10 enzymes. And then if you get to 20 enzymes and 50 enzymes and 100, again, the enzymes are just waiting for a substrate to come along. So you can see that there becomes another limiting factor. So that's when it plateaus. Okay, Slido poll. So I'll skip the one about ethics. So reactions involving enzymes will not happen if the enzyme is not present. True or false? Yeah, okay, again, for the sake of time, sorry to cut you guys off a little bit. But yeah, true. The answer, correct that the answer is false, which most of you have said. So yeah, remember this idea, enzymes speed a reaction up. So the reaction will happen very slowly, but it will not stop. I remember this because this is something I got wrong in one of my SACs, I think. That idea that to be technically accurate, if you don't have an enzyme, the rate will be so slow, it'll basically seem like the reaction isn't happening, but it just is happening really, really slowly. An enzyme does not turn a reaction on or off, it just speeds it up. Okay. So we'll go through photosynthesis as quickly as we can. So the idea we're thinking of here is we need to get an energy source for the plant. So we're using light energy and we're turning it into our glucose that our plant can use basically to produce ATP so that they can grow as well. It's really important. All the time I think I see this misconception that photosynthesis only in plants, cellular respiration only in animals. Cellular respiration is in plants as well. They need ATP, they need to grow. It's just that photosynthesis is only in plants. So don't kind of isolate the two completely. Okay, so it's happening in our chloroplasts. So these are found in plants as well. So specifically kind of in our like leafy cells. So our chloroplasts, we've got our 2 important structures. So the grana and the stroma. So the grana or a granum is made up of thylakoid discs. And this is where our chlorophyll sits. And that's really important in capturing the light energy. And that's why it's green. And so that is where we have our, think chlorophyll, green, our grana, light-dependent reaction. Stroma is our, just like liquid that floats around in here. Think of it as like the chloroplast version of the cytosol. And that is where our light-independent stage happens. Okay, so here's a bit of a better diagram. So we've got our light-dependent and we have our light-independent. This is a really nice, good overview of the process. So we'll talk about the steps, but basically your water comes in. We've got our light energy that's giving us the energy to produce this glucose essentially. The reason we want water is for the hydrogens. The plant couldn't care less about the oxygen. The oxygen gets sent out. These hydrogens come over into the light independent stage. And how they do it is they come over via the NADPH. Remember when we talked about coenzymes? So we've got like ATP, our NADPH, stuff like this. So importantly, remember the coenzyme is NADPH. In cellular respiration, you've got NADH, photosynthesis, P for photosynthesis, P, NADPH. So we need our hydrogen that's popped over here. Basically, the role of NADPH, our coenzymes, is just to cycle. Again, coenzymes, they act like enzymes a little bit. They're not like used up themselves. They're just little transport vesicles almost. And that's kind of what our ATP is used for as well. Photosynthesis, we don't have any net ATP production. We get some ATP from our light energy and we use it in order to produce our glucose. So it's just that cycle. So our carbon dioxide comes in the light independent stage and fuses with our hydrogen, and therefore you get glucose. So that's a really nice rough overview. To be a little bit more specific, again, so our water molecules, you can see, split our hydrogen and our oxygen. Oxygen is our waste product. Our hydrogen goes to the next stage via our NADPH. Our ATP is produced going into the next stage as well. This is in the grana. The light independent stage is in our stroma, so in this liquid. It's also known as the Calvin cycle. And so we use our ATP, we use our NADPH, they are all cycled. Our carbon dioxide comes in and we then produce our glucose. So you can see that oxygen is basically produced and released in the light dependent stage. It doesn't really have any purpose over here. Our carbon dioxide isn't involved in the light dependent stage at all. It only comes in here. So understand that. Rubisco is also an enzyme that we look at in particular that's involved with the Calvin cycle. And it's important in this idea of carbon fixation. So here are our inputs and our outputs. Quick Slido poll. So, which molecule is not involved in the light dependent stage? So I know that was a bit of a quick overview. Hopefully that makes sense. Again, just like for me, visually, it's really important to kind of let everything sink in. It does depend on the type of learner you are. But yeah, diagrams for cellular respiration especially, because there's 3 stages, they really help a lot in my opinion. So really, really work with them. So light dependent, remember what happens in the light dependent. Think of what splits in the light dependent, what is involved there. Okay, lovely. So yeah, correct, carbon dioxide. Remember this is involved in our light independent stage. So with the stroma, with the Calvin cycle, oxygen gas is involved in the light dependent because water splits to form our oxygen gas and that is released. ATP, remember that's produced in the light dependent stage and used up in the independent stage. And water, that's kind of our main input. So basically know your inputs and outputs. So the main input of the light dependent is the water, main output oxygen, main input of light dependent is carbon dioxide, main output is glucose. Understand that, and then the other bits kind of fall around. This is what I meant when I was answering the question before about how to know your complex details and learn that information. Break it down to your basics first, so kind of what I've just mentioned, main input, main output, main input, main output, and then add the other stuff in, the location, the coenzymes, all that kind of other stuff. Okay. So in terms of Rubisco, again, just a brief overview. Basically, Rubisco, again, all comes down to stuff with enzymes. Rubisco is an enzyme, so all the stuff that we talked about before applies here. So essentially, Rubisco, its substrate is carbon dioxide. Oxygen looks very similar to carbon dioxide. So what happens is Rubisco will obviously work on carbon dioxide. When carbon dioxide is low, it will bind to oxygen because they look similar. You don't want that. You want to avoid Rubisco binding to oxygen at all costs because as you can see here, basically, yeah, everything kind of slows down and it wastes resources, it wastes energy. And yeah, you basically just want to avoid that. So C3 plants, they're your really basic plants. It's what I've just talked about in terms of photosynthesis. That's how it happens. They don't have any way of preventing Rubisco from binding to oxygen. However, C4 and CAM plants, they basically do. I don't know. Yeah, I don't think we'll go into it. But basically, C4 separates it by space and CAM plants were separated by time. That's kind of the main thing you need to know. So again, I won't go into it in too much detail, but CAM plants separate it by time. So it's this idea that it's almost like, I think of it like hibernation, like when a bear eats a lot of food and then sleeps, like it's kind of like that. You store the carbon dioxide at night to use it in the day. In C4 plants, basically they store carbon dioxide in this area of the plant to use it in another area. So that's the general overview there. Okay, so basically the exact same thing that we talked about with our enzymes. So with light availability, we've gotten our plateau essentially. So the more light you add, that's going to increase the light dependent stage, which in turn will increase light independent. You're getting that increased rate of photosynthesis. Again, it plateaus. It'll always plateau because something else will become the limiting factor. You can add as much light as you want, but ultimately, there's only so many enzymes in the plant, or there's only so much water that you have, or only so much carbon dioxide. Something else becomes the limiting factor. Same with water availability. So the more water, again, water is an input of our light dependent stage. It increases. You get to that plateau. Carbon dioxide concentration, the same thing. It increases. You get that increased rate of the light independent stage. Ultimately, something else becomes the limiting factor. You can add as much carbon dioxide as you want. The light is the limiting factor. Light energy can't work any faster. Hopefully that makes sense. See how it's the same principle as what we've talked about before? So work smarter, not harder. Memorize one, understand that concept, and then just apply it. Okay, so moving on to cellular respiration now. So I think, in my opinion, cellular respiration is a little bit more complex than photosynthesis, but I don't know, it can depend. So basically, we're doing the reverse now. So instead of using our energy to produce glucose, we're breaking down our glucose to produce our energy. So we have aerobic and anaerobic. Aerobic, we need oxygen. We produce a lot of ATP, and we need the mitochondria. In our anaerobic respiration, it happens super duper quickly, but we don't produce as much ATP. Basically, it's just happening so much faster that we're producing less ATP within the process itself, but because it's so quick, we just can get like a big bang of ATP out. Don't need the mitochondria, obviously doesn't have oxygen. So these are our 3 steps. I will kind of preface this by saying this cellular respiration can be very complex to kind of understand. I know I feel like I've said this with every single process, but it does take time. Have a general overview. Diagrams are your best friend. The more you revise it and revise it and revise it, one day, like, it'll just click. Okay, so this is the first step, the general overview. We're not into the mitochondria yet. We're just breaking down the glucose. Again, I can't emphasize this enough with cellular respiration. This is probably the most important of all the processes in terms of bigger picture. Breaking it down and understanding what is the process, because there's so many kind of intricate details. It really helps if you can understand what is our main objective of glycolysis, what is our main objective of Krebs cycle, what is our main objective of ETC. So our main objective of glycolysis, it's in the name, glycolysis. We're basically splitting glucose. We're doing that initial breakdown into a bit of a more simpler molecule. So this is in the cytosol. It happens in anaerobic and aerobic respiration because it doesn't require oxygen, doesn't require our mitochondria. So we've got our glucose molecule. You'll often see it kind of referred to as how many carbons are in the molecules. But basically glucose has 6 carbons, right? C6H12O6. So we get our 1 6-carbon molecule and we break it into 2. So now I've got 3 carbons, 3 carbons. These 3 carbon molecules are called pyruvates. Hopefully that makes sense. Simple enough. We're just literally splitting that glucose because remember, the whole objective is we're breaking it down to get energy. So this is just the first step of breaking it down. We do get a little bit of, let's just say ATP, not ADP. We get 2 molecules of ATP, and then we have NADH as well. Remember, not NADPH, NADH. So I've got our ATP and our NADH. That's it for glycolysis. Relatively simple, at least compared to the other 2. So now going into the Krebs cycle, you have this kind of intermediate reaction or this early thing, and that's just converting pyruvate into acetyl-CoA. So this technically isn't the cycle yet, but we have pyruvate, which is our 3-carbon molecule, and we convert it into acetyl-CoA, which is our 2-carbon molecule. So we have pyruvate that goes to acetyl-CoA. The other carbon is CO2. So remember, CO2, we produce CO2. It's a product of cellular respiration. So one molecule kind of comes from that conversion. We also have a bit of NADH as well. Step 2, this is where kind of the actual, money of the reaction is. So we are now in the mitochondrial matrix. So this is all happening in the mitochondria. Krebs cycle and etc. It's in the mitochondria. So the mitochondrial matrix, it's basically the fluid. It's kind of the mitochondria equivalent of the stroma. So we have the Krebs cycle going on, and the acetyl-CoA basically goes in, and we get all of our coenzymes. So we get CO2, that's a bit of another thing. But importantly, we get some ATP, and we get our NADH and our FADH2. These are really important because we're going to use them in the ETC. Remember, this is what I'm talking about with purpose. So purpose of glycolysis, to split glucose into our pyruvate, so our early breaking down. The purpose of Krebs cycle is basically to load these coenzymes so we can use it in ETC. ETC is our nice main objective of cellular respiration, and it's where we're going to get our huge burst of energy and our huge production of ATP. So the Krebs cycle is basically just getting the ingredients ready to put them into the ETC. So we need our NADH and we need our FADH2, and we've also got a bit of ATP and CO2. You might think in total, the Krebs cycle produces 2 molecules of ATP. So we get double this, because remember we had 2 pyruvate molecules to start off with. So then we're going to get 2 acetyl-CoA molecules. So we get double all of this. Honestly, the numbers aren't really important. I don't think I've really ever seen a question like how many FADH2 molecules are appearing. So I would say just remember it if you can, but it's not the biggest thing to be aware of. The actual names themselves, so knowing FADH2, knowing NADH, knowing your CO2, etc. That's the really important stuff. Okay, now we get into the main gist of it. So this is the cristae, sorry, spelled like this, cristae. So this is our inner membrane of our mitochondria. So remember we were in the matrix, which is the fluid that's in that, kind of bound within the cristae. Now I'm moving to the actual membrane itself. So essentially it can be very confusing. You do just have to break it down. Again, kind of try to think of the purpose. Watch as many videos as you need to, look at as many diagrams as you have to, and importantly, understand when you're going beyond the level of what you need. So essentially we have our electrons. So remember that we have NADH, we've got our FADH2, they're carrying our little hydrogens, right? So our hydrogens, basically they will give out electrons, hence electron transport chain, and these will be transported along the membrane. So we have our electrons, they move through, you can see just moving along. And then our hydrogen kind of moves along as you saw there. I think that's it. Yeah. Okay. So I'll talk about, the electrons first. Essentially, they just move through, and they're literally just transported and they're carried through. The hydrogens, as you can see, are pumped over the other side of that membrane. So the intermembrane space, that's where they're kept. And again, I don't want to get too technical with this, but they get shoved into the intermembrane space, and this creates this kind of force, what's called a proton motive force, that basically gets our ATP synthase running. ATP synthase, the enzyme that makes our ATP. So we have our electrons that are being carried along, our protons, so our hydrogen ions, they're being shoved into this space, and they're basically pushing this ATP synthase down. So you can see that here. So a kind of analogy that helped me think about this was, I got told this in uni actually. It was the hydrogens, if you can think of pushing these hydrogens, it's pushing water up a hill and then having it come down, almost like a dam, or getting, those wheels that spin with the water, a hydro, I'm thinking of, I don't know, hydroelectric plant, whatever it is. This idea of pushing that water up and then it's coming down and then it's producing that energy, if that makes sense. So yeah, that analogy kind of helped me a little bit. I will say VCAR doesn't really need that technical of an explanation. I think it just does help to kind of wrap your head around it, because even before I sort of heard that analogy, and I would always kind of see this stuff in textbooks, but it was never really explained. in my head, I used to just be like, oh, I don't really know what goes on in the electron transport chain, but I know somehow like ATP synthase is turned on. So basically, I'm just trying to explain how that works. But again, you wouldn't really be expected too much to write about like, you know, hydrogen ions or proton motive force or stuff like that. Generally, you just need to know that the electrons will be carried along this chain. ATP synthase will basically produce your ATP. And importantly, your hydrogens, they will basically mesh with your oxygen and that will create water. And then there's our output. Hopefully that makes sense. Again, I know that is really tricky, but try to think about that a couple of times. Try to let it sink in your head. But yeah, so basically we've got our NADH and our FADH2, so our coenzymes. Their purpose is literally just to carry our electrons and our protons. So they give their electrons, passes along here. The the protons get shoved into the intermembrane space. This causes a force that basically pushes our ATP synthase to get working, and this is where all our ATP comes from. This is the main purpose of this whole process. And then our protons are left over, and they're like, where can we go? They combine back with their little electron, and the oxygen that we're breathing in is waiting there to collect those hydrogens and to form water. Hopefully that makes sense. Okay, this is a really nice diagram that kind of captures that. So as you can see, it's 30 or 32 ATP because in the electron transport chain, you're producing 26 or 28. And so plus the initial 4, that's what you're getting. So again, understand this was kind of specified by VCAR with last year's study design, so the new one. In old resources, you'll see that it is 36 or 38. So just be mindful of that. Okay, Slido poll. Oh, okay, wait, I think this is the last one. No. Okay, sorry, wait, maybe I'm missing a Slido poll. Am I not looking properly? Oh, okay, sorry, it's been swapped around. How many ATP molecules are produced in the ETC? Okay, so something I've literally just mentioned. So try to think about it. So think about the question. How many ATP molecules are produced in the electron transport chain? Okay. I know we do want to get into answering some questions soon, so I'll stop the voting there. Yeah, 26 or 20, or did I stop it? Yeah, 26 or 28. So that is in the ETC, plus our 2 from the Krebs cycle, plus our 2 from glycolysis. That means the overall of aerobic respiration is 30 or 32, but in the ETC itself, 26 or 28. So yeah, just be mindful of that. Okay, so again, it's the exact same thing. So you're looking at temperature, it's the same. Glucose availability, it is a substrate. Glucose is the substrate, so you're getting that plateau. Oxygen concentration, it's an input, you're getting that plateau. The only thing to be aware of is with oxygen concentration, like we're thinking about switching from aerobic to anaerobic as well. Okay, so anaerobic respiration, we're looking at, again, basically like a really simple pathway. This is when oxygen is low, when we're in a low oxygen environment, that we're still able to get ATP produced, essentially. So glycolysis, exactly the same, breaking into our pyruvate. We've got our 2 ATP. This is literally it. That's where the ATP comes from. This process just happens so quickly that it just goes 2 ATP, 2, 2, 2, 2, 2, 2, 2, rather than having to wait a bit of a long time to get 30 or 32 from the longer process. It just happens very quickly so that we can just get it real quick, really. So from there, the pyruvate is either converted into lactate or lactic acid in animals, and in yeast, it's converted into ethanol and carbon dioxide. That's the main thing to know. Like the NAD, stuff like that, I don't think I even really knew that. The number, I don't think I even knew that as well. Just understand it's either lactic acid / lactate in animals or your ethanol and your CO2. And yeah, cellular respiration, photosynthesis are the most commonly tested for that like scientific poster for area of study 3, SAC. So know these processes really well. Yeah, and it's in the cytosol, by the way. Okay, sorry for that. Yeah, so essentially that's it. I know that was so quick.